my name is Hannah Miller, and today I'll be presenting on the paper entitled Hypoxia-Inducible Factor 2-alpha as an Essential Molecular Brake for Postprandial Hepatic Glucagon Response Independent of Insulin Signaling. This paper was written by Sadish et al. In order to understand how this hypoxia-inducible factor works, it is important first to understand and have a background on glucagon signaling. As you can see in this image, glucagon is secreted when the body senses low blood glucose levels. This hormone attaches to a G-coupled protein receptor, which then activates adenyl cyclase. Adenyl cyclase, in turn, increases the concentrations of cyclic AMP in the cell, which activate pKa and an enzyme cascade. Ultimately, this enzyme cascade ends up activating the transcription factor CREB, which is responsible for upregulating the transcription of gluconeogenic enzymes such as G6P and PEP carboxykinase. Glucagon, under normal conditions, drives gluconeogenesis, maintains blood glucose storing fasting, and maintains homeostatic blood glucose levels after absorption. When glucagon is dysregulated, it can result in hyperglycemia. Hyperglycemia is very common in type 2 diabetes, and this is why the authors had the interest to study hypoxia-induced factor 2-alpha, which is known to attenuate glucagon. Insulin is known to inhibit glucagon via a deactivation of the CRTC2 transcription factor. This method, however, involves insulin in the IRS and pKa pathway. The authors wished to avoid using insulin, and so were very interested in this hypoxia-inducible factor. The hypoxia-inducible factor was first studied in patients with Chuvis polycythemia, a congenital oxygen sensing disorder. This disorder was known for a homozygous mutation in the von Hippel-Lindau protein, the VHL protein. This protein is known to degrade hypoxia-inducible factor. One of the main symptoms of people with Chuvis polycythemia was hypoglycemia. This was very interesting to the authors, and so they studied it more. Hypoxia-inducible factor under normal conditions is usually stabilized by hypoxia in the liver. Otherwise, it would be degraded by VHL. Past studies have shown that hypoxia-inducible factor 2-alpha increases insulin sensitivity and improves glucose homeostasis via insulin signaling pathways. But this study desired to determine its role specifically in insulin-independent glucagon regulation. In order to study the insulin-independent mechanism of hypoxia-inducible factor 2-alpha, the researchers used mice and disrupted their VHL gene. The researchers broke up their results into 7 different sections. In the first section, mice were treated with tamoxifen-inducible Cray recombinase under the control of the albumin promoter to disrupt their VHL in the liver, and compared to then wild-type mice with no disruption. Insulin, glucose, and glucagon tolerance tests were performed on the mice, in addition to testing the expression of G6Pase and PEP carboxykinase, which are key in the gluconeogenesis pathway. As seen in Figure 1, mice with a disrupted VHL gene exhibited lower fasting glucose levels and gradual glucose tolerance, as seen in A and B. The improved glucose homeostasis in the disrupted mice is not due to increased IRSS2 insulin signaling in primary hepatocytes, as no difference was observed between the insulin signaling pathways of the disrupted mice versus the non-disrupted mice. This can be seen in Figure 1C. A PKB inhibitor, wortmannin, did not restore expression of G6Pase or PEP carboxykinase in the disrupted mice. The disrupted mice exhibited increases in FOXO1, a critical regulator of gluconeogenesis, thought to circumvent hypoglycemia. Upon fasting, the expression of PEP mRNA was significantly attenuated, and G6Pase was completely restricted in the disrupted mice. A significant decrease in serum insulin levels and an elevation of plasma glucose levels, Figure HIF2a, and 1J, led the authors to conclude that the decrease in hepatic glucagon action may be responsible for improved homeostasis in the VHL-disrupted mice. Next, hepatic gluconeogenesis was assessed by a pyruvate tolerance test in the disrupted mice. These mice were tested at 1 and 2 weeks after the disruption. Later, primary hepatocytes were treated with PKB inhibitors and an AKT inhibitor. Disrupted mice exhibited both a significant decrease in hepatic G6Pase activity with an increase in glycogen content as seen in figure 2b and 2c. Also, a decrease in both basal and glucagon-induced glucose production. Glucagon's induction of G6Pase and PEP carboxykinase was completely abrogated in the disrupted mice. Inhibiting the insulin signaling pathway through a PKB inhibitor and an AKT inhibitor did not restore glucagon induction of G6Pase mRNA or hepatic glucose production as seen in figure 2G and 2I. The mechanism, therefore, for the blunted glucagon action and decrease in glucose output was a result of the attenuated glucagon Krebs signaling in the liver. Next, in order to determine whether it was HIF1 alpha or HIF2 alpha which was mediating the decrease in hepatic glucagon, mice with a combined disruption of VHL and HIF1 alpha were compared to mice with a disruption of both VHL and HIF2 alpha. Lower blood glucose levels of the disrupted mice were reversed by the disruption of both VHL and HIF2 alpha, but not by the VHL and HIF1 alpha, as seen in figure 3a. The mice with the combined disruption of VHL and HIF2 alpha exhibited increased plasma insulin levels and normalized serum glucagon levels, as seen in figure 3b. Yet there was no observable difference in insulin sensitivity. The researchers determined from this data that the improvement in glucose homeostasis and attenuated glucagon action in the VHL-disrupted mice is HIF2 alpha dependent. In order to determine the mechanism by which HIF2 alpha represses glucagon signaling, the researchers treated the primary hepatocytes of the disrupted mice with a phosphatase inhibitor, OKA. OKA treatment did not restore glucagon-stimulated Krebs phosphorylation or G6Pase in the disrupted mice, as shown in figure 5a. Combining pKa once again in the disrupted mice did induce Krebs phosphorylation. The authors therefore concluded that pKa activity must be repressed in the disrupted mice. Specifically, this disruption was in the R2 subunit. When treated with glucagon, the wild-type mice exhibited a 10-fold increase in the intracellular cyclic AMP levels, while in the VHL-disrupted mice, this response was significantly attenuated, as shown in figure 5d. Adding an adenyl cyclase activator had no effect on the Krebs phosphorylation. When more cyclic AMP, however, was added, there was complete restoration in the pKa and Krebs signaling, as shown in figure 5f. This allowed researchers to conclude that HIF2 alpha represses glucagon pKa signaling by reducing the cyclic AMP intracellular concentrations. Since the authors knew that intracellular cyclic AMP levels are regulated by phosphodiesterases, they assessed the phosphodiesterase activity specifically to further study how the hypoxia-inducible factor 2 alpha repressed glucagon signaling. Phosphodiesterase activity in glucagon repression was assessed through the addition of many different activators and inhibitors. Hypoxia-inducible factor 2 alpha was found to regulate PDE4 specifically through post-translation modification, as there was no observed increases in PDE4 mRNA or protein levels. To outline the pathway of HIF2 alpha's regulation of glucagon signaling upstream from PDE4, the researchers screened for glucagon-induced G6Pase expression in the wild-type mice and in the VHL-disrupted mice in the presence of many activators and inhibitors. After much screening, inhibitors of MEK and ERK were found to partially restore glucagon-induced G6Pase expression. This data gives evidence that ERK is a critical regulator of hepatic glucagon signaling in the VHL-disrupted mice. Additionally, the authors concluded that HIF2 alpha transcriptional activity is essential for ERK regulation of gluconeogenesis, and that HIF2 alpha represses the glucagon signaling partially by activating ERK signaling. In order to test the effects of hypoxia-inducible factor 2 alpha in diabetic mouse models, the researchers designed a mouse with a temporary disruption of hypoxia-inducible factor 2 alpha and compared it to littermate controls. All these mice were fed a high-fat diet for 6 weeks, and then their glucose and insulin tolerance levels were tested. No difference was observed between the glucose and insulin tolerance tests, but the mice with the disrupted HIF2 alpha did exhibit an increase in glucagon response, as seen in Figure 7c. This shows, along with previous studies, that hepatic HIF2 alpha increases insulin sensitivity and dampens glucagon response. Since it is known that the hypoxia-inducible factor 2 alpha improves diabetic parameters in diet and genetic-induced obesity through insulin signaling pathways, insulin-resistant induced diabetic mice were used in this experiment, and then their VHL gene disrupted. Blood glucose levels increased for all the mice, but by day 6, the glucose levels in the VHL disrupted mice returned to normal, demonstrating that the disruption of the VHL improves glucose homeostasis in both insulin-deficient and insulin-resistant animals by repressing the hepatic glucagon response. In summary, the hormone glucagon plays a critical role in maintaining glucose levels during fasting by activating metabolic pathways such as gluconeogenesis and glycogenolysis. This work focused on studying the mechanisms which regulate glucagon signaling, specifically those independent of insulin signaling pathways. A temporary postprandial increase in hypoxia-inducible factor 2 alpha was revealed to be a critical repressor for glucagon signaling. During fasting, circulation is concentrated towards the liver, but after refeeding, it is redirected towards the small intestine. Hypoxia-inducible factor 2 alpha is stabilized by this redirection of circulation and the temporary hypoxia in the liver. When hypoxia-inducible factor 2 alpha is stabilized, it suppresses glucagon signaling by increasing phosphodiesterase activity and by increasing cyclic AMP hydrolysis in an MEK and ERK dependent pathway. Further research must be performed on the integration of glucagon and insulin signaling by hypoxia-induced factor 2 alpha, as all the pathways and mechanisms are still not fully known.